Hello, this is Mr. Yan and welcome back. This is video 2 on viruses, and if you remember from video 1, we concentrated on the structure of viruses. And for this video, the big question is, how do viruses infect cells? And if you can describe that process by the end of the video, you're doing well. Also, some chance for some extra credit. If you can describe how a virus is like a Trojan horse, just write that, your description on a half sheet of paper, turn it in tomorrow, and you'll get some extra credit for that. If you know what the Trojan horse is, this might be a bit easier for you, but if you don't, look it up. It's worth knowing. So again, how do viruses infect cells? And let me go back to my Painter program and show you some illustrations of this process. First of all, some review. Well, this is the virus right here. And if you notice, the virus is a lot smaller than a typical cell. And this is the cell that the virus is going to infect. And again, the virus has a protein capsid. Inside this capsid is its genetic material, either DNA or RNA. And like a cell, a virus can be surrounded by this lipid envelope with these proteins sticking out. And I mentioned these blue proteins right here are sort of like keys, and they can unlock the lock of certain cells. And it just so happens that cells have proteins sticking out as well. And if these two proteins were to match up, then the virus can gain entry into the cell. And also notice that, this virus is sort of acting like a cell. It's in disguise. The cell doesn't really want the virus in here, so the virus has to fool the cell somehow into gaining entry, and this is how it happens. Here's an illustration, a blown up illustration showing you the detail. You have a cell right here, part of a cell, the cytoplasm is right here. You have another cell with its cytoplasm, and if you notice, these cells have different proteins on their cell membranes, and they have, in particular, different shapes. Here is like an arrow shape, here is a circle shaped, and this is very simplified, but it's still giving you a good idea of how this process works. Now, ask yourself, these proteins, are they going to match up better with these proteins or this? And I think the answer should be pretty obvious, that the proteins for the virus are going to match up with the proteins on the surface of the cell. So this would be the virus protein right here, and it would match up very nicely, dovetail right in there with the cell's protein. And if this happens, if the virus comes in contact with a cell like that, the cell is sort of fooled into thinking that it's letting a friendly particle inside the cell. The cell has no idea it's letting in an enemy, which is the virus. Whereas over here, this virus protein, undo, this virus protein is not going to match up very well with that circle on top of this cell's protein. So that's why some viruses why viruses infect certain cells and not others. Okay, so if the proteins on the surface of the virus and the cell actually match up, this is what happens. The cell allows the lipid envelope of the virus to actually fuse with the cell membrane of the cell, and this lipid envelope right here of the virus actually becomes part of the cell's membrane. And in the process, this virus can enter the cell. And you will see this is a lot like a Trojan horse, and if you can explain that process, you can get extra credit tomorrow. Another way that a virus can enter the cell is in the case of these bacteriophages. Bacteriophages are viruses that infect bacteria, so this would be my bacteria cell right here. Bacteria, change color. And this is my virus. And notice this virus doesn't have a lipid envelope. Some viruses don't do this. This virus in particular is going to be able to poke its way through this cell wall and cell membrane of the bacteria and actually inject its DNA into to the bacteria itself. It will inject its DNA into the bacteria. And even though this protein capsid doesn't get in there, that's all that's important for the virus, is that its DNA gets in there, and now the cell has instructions to produce more viruses. So that's an alternate way that viruses can enter their information inside a cell as well. Now let's go back to my PowerPoint. So this objective, describe how viruses gain entry into cells. I just described all that stuff. And for your fill-in notes, you should be able to get most of the information from this slide right here. And I'm not going to go over it again, but you can pause the video right here and copy down information if you need more time. And here is some more information that might be relevant for your notes as well. And again, you can pause this slide and copy if you wish. But moving on, here is another electron microscope produced image of viruses. And this time we have several viruses right here. And these viruses are interacting with the cell membrane. And I'm not really sure what's happening right here. It could be one of two things. These viruses might be interacting with the proteins on the membrane, trying to gain access into the cell, which looks like might be happening because look, these viruses are whole inside the cell. Or alternatively, these viruses might be poking little holes through the cell membrane and injecting their viral DNA into the cell. And as you'll find out in the next video, whatever the case, the same result happens every time. The next video is going to be on what happens once this DNA or RNA or the virus itself gets inside the cell. And I will see you next time.